Overview of photosystem I. Function: sees an integral membrane protein complex responsible for catalyzing the transfer of electrons across the thylakoid membrane from plastocyanin to ferredoxin. The transferred electrons are used to produce the hydrogen carrier NADP, and the photon energy absorbed generates a proton motive force to produce ATP. History: discovered before photosystem II, it was later recognized that photosystem II is the first enzyme of the photosynthetic electron transport chain. Louis Dysons and others proposed the concepts of photosystems I and II in the 1960s. Components and action: main subunits Sa and Sab are involved in binding essential electron transfer cofactors. More than 110 cofactors, including iron-sulfur clusters F, Va, F, ferredoxin, plastocyanin, and pigments chlorophyll A, carotene. Antenna complex with chlorophylls and carotenoids for light absorption. P70 reaction center absorbs light at 700 nanometers, initiating electron transfer. Evolution: silically evolved from the photosystems of green sulfur bacteria, showing similarities in redox potential, iron-sulfur proteins, and protein subunit dimers. Key protein subunits Sa and Sab, largotransmembran proteins binding essential cofactors. Integral membrane proteins with 11 transmembrane segments. SAC iron-sulfur center binds to the Sa or Sab core near Fx. Other subunits SAD, C, Psi, SOJ, SAC, Cell, SAM, SAX, and cytochrome B6F complex. These subunits have various functions, including assembly, stabilization, and binding of electron carriers. Electron transfer chain antenna complex, composed of chlorophylls and carotenoids transmitting resonance energy from photons. P700 reaction center monophecally absorbing light at 700 nanometers. Initiates electron transfer and forms a dimer with an electric potential. Electron acceptors monophecally into 1, phylloquinone, iron-sulfur complex F, Va, F, ferredoxin, and plastocyanin. Ferredoxinad plus reductase for enzyme facilitating the transfer of electrons from reduced ferredoxin to NAT+. Evolved in a recipe, silically evolved from a common ancestral photosystem shared with green sulfur bacteria. Importance of YCF for protein domain, Phe for protein domain found on the thylakoid membrane, is vital for assembling the components of photosystem I, contributing to efficient photosynthesis. The detailed description provides insights into the structure, components, and functions of photosystem I, shedding light on the intricate processes involved in the photosynthetic light reactions.